MStem laboratory started about 5 years ago when we started to obtain embryos, human embryos, donated to the University of Michigan to make human embryonic stem cells. We make human embryonic stem cells and then make those cells available to researchers at the University of Michigan, throughout our state of Michigan, but also across the country, where they can actually use those embryonic stem cells to study specific diseases.
This has allowed researchers like myself, who's a clinician and a scientist who's interested in doing research on specific diseases, to gain access to a new model system that can be very powerful in studying aspects of human disease. Fragile X syndrome is the most common cause of intellectual disability and autism. So our lab is interested in trying to figure out ways that we can better understand that disease and treat it.
Any of the experiments that we do and the conclusions that we reach in the lab are really limited to the model system we're working with. We're trying to recapitulate what happens in a disease here in the lab so we can study it and we can treat it, and hopefully that will translate into people. But all of that really depends on how accurate your model system is. That's the real benefit of using stem cells, because these come from the people with the disease themselves. There's no other better model. And so we think that by using these cells, we can more accurately see what's happening in disease, and the things that we identify in the lab that help should also be able to help in people.
We have modeled a condition called hypertrophic cardiomyopathy using these embryonic stem cells, and we can study the cardiac myocytes that they create and how they beat and what they look like and how they're aligned and how they interact with other cells next to them. What's really powerful is that we can use them to study disease in a particular patient with a particular genetic mutation, and can compare them to other models that have different mutations or other patients with the same mutation.
The origin of MStem really takes its roots in human IVF, and the years of experience that I had and many of the people in our laboratory in working with human embryos and in treating infertility. We've actually used that expertise to be efficient in deriving human embryonic stem cells from these embryos that are donated to us. These couples make this decision about embryo donation, and they don't do it lightly. And we feel that it's really important that we give the best effort of using these human embryos to make an embryonic stem cell line. And that in itself is really what's going to help in the treatment and the cures of many of these single gene disorders.